So let us continue about mechanism of cell injury. Today we will be discussing the damage to DNA and uh, free radicals. So damage to nuclear DNA will activate these sensors that will trigger P53 dependent pathways. Okay, so DNA damage will occur due to radiation, chemotherapeutic or consumption of anti-cancer drugs, due to interaction of reactive oxygen species, or may occur due to aging, or also mainly due to deamination of cytosine residues to uracil residues. So DNA damage will activate P53. So what is the function of P53? Is it will arrest the cells in G1 phase of the cell cycle. Okay, and it activates the DNA repair mechanisms. In case if the DNA cannot be repaired, then P53 will trigger apoptosis. So there are two possibilities. Since P53 will function of P53 is to arrest the cell in G1 phase, it will either activate DNA repair mechanism. If the DNA fails to repair, then P53 will trigger apoptosis through the mitochondrial pathway. Okay, so the cell will chooses to die rather than surviving with abnormal DNA. Thus, it has the potential to produce malignant transformation. So, if the DNA cell with abnormal DNA survives, then it has the capability to produce malignant transformation, leading to tumors. So, mutations in P53 will interfere with its ability of the cell to arrest cell cycling or to induce apoptosis. Okay, so next we will see about oxidative stress, that is accumulation. What is oxidative stress? The accumulation of oxygen derived free radicals. So cell injury which is produced by free radicals, particularly reactive oxygen species, is an important mechanism of cell damage in many pathology conditions. See, in case of chemical or radiation injury or ischemia reperfusion injury. What is ischemia reperfusion injury? So it is produced by restoration of blood flow in ischemic tissue, or due to cellular aging and microbial killing by phagocytes. Free radicals are generated. So free radicals are chemical species having single unpaired electron in an outer orbit. So these unpaired electrons are highly reactive and attack and modify molecules to inorganic or organic chemicals like proteins, lipids and carbohydrates, nucleic acids, many of which are important components of cell membranes and nuclei. So they destroy the components of cell membranes and nuclei. So these reactions are usually autocatalytic, that is, molecules reacting with free radicals or themselves converted to free radicals. Thus, it is similar to nuclear decay, that is, one atom will trigger the other atom and so on. So samely, any chemical or any molecules that will react with free radicals will, that molecule itself is converted into free radicals. Thus, it will increase and propagate the chain of damage. So, ROS, reactive oxygen species, are usually produced in cells during mitochondrial respiration and energy generation, but they are degraded by ROS scavengers, as the name indicates. So, this defense system will allow the cells to maintain a steady state in which free radicals may be present at low concentration. So, we want free radicals to be present at low concentrations, and so that they do not cause any damage to the cell. For that, we need ROS scavengers. So, first is oxidative stress. So this is very important. Oxidative stress means like increased protection or decreased protection of ROS, or decreased scavenging of ROS may lead to huge quantity of free radicals. That is what called as oxidative stress. So oxidative stress is seen in many pathologic processes including cancer, cell injury, aging and Alzheimer's disease and so on. Usually, ROS is produced in large amounts by activated leukocytes, so particularly neutrophils and macrophages, especially during inflammatory reactions which are aimed at destroying microbes and cleaning up dead cells and other unwanted substances. Okay, so next we will be seeing about generation of free radicals. So free radicals are usually generated in several ways. First one is reduction oxidation, that is redox reactions, which will occur during normal metabolic processes. So molecular oxygen is reduced by the transfer of 4 electrons to H2 to generate 2 water molecules. So oxygen is reduced by transfer of 4 electrons to hydrogen molecule to generate 2 water molecules. So this conversion is catalyzed by oxidative enzymes in the endoplasmic reticulum, cytosol, mitochondria, peroxisomes and lysosomes. So during this uh, process, small amounts of partially reduced intermediates are produced. So during the formation of two water molecules, the steps involved in this formation involves different number of electrons have been transferred. Intermediates are produced containing different number of electrons that have been transferred from oxygen. So these may be superoxide anion, which has one electron, that's a free radical, and hydrogen peroxide 2 electrons, and hydroxyl radicals having 3 electrons. Next, due to absorption of radiation, so ionizing radiation will automatically hydrolyze water into OH and hydrogen free radicals. Next, in activated leukocytes, as you see during inflammation, a huge quantity of ROS is produced uh, because to destroy the injured cells. So in a precisely and its cells, this occurs in a precisely controlled reaction, which are carried out by plasma-membrane multiprotein complex that uses NADPH oxidase for the redox reaction. So during inflammation, this produces. So if you remember that is enough. So defect in leukocytic superoxide production leads to chronic granulomatous. We'll be seeing in next chapters. Next, enzymatic metabolism of exogenous chemicals or drugs. Okay, so enzymatic metabolism of the drugs can generate free radicals which are not reactive oxygen species but have similar effects. For example, C scale for carbon tetrachloride can generate carbon trichloride free radical, and transition metals. If we take transition metals, iron and copper can donate or accept free electrons during intracellular reaction and catalyze free radical formation. So in Fenton reaction, hydrogen peroxide combines with ferrous ion to form ferric ion, and ferric ion one free radical of hydroxide and one hydroxyl ion. So most of the intracellular free ion is in the ferric state. It must be reduced to ferrous form to participate in the Fenton reaction. So Fenton reaction is nothing but conversion of ferrous to ferric ion in presence of hydrogen peroxide. Usually in our body cells, free ion is nothing but ferric state, but it must be reduced to ferrous before it can participate in the Fenton reaction. Next, we will see a nitric oxide. So nitric oxide is an important chemical mediator generated by endothelial cells, macrophages, neurons and other cell types. So even this is acts as a free radical and can be converted to highly reactive peroxynitrite anion, as well as NO2 nitrite and nitrate anion, hydrogen dioxide and nitrate anion. So removal of free radicals. So you see how free radicals are removed. Free radicals are usually unstable and generally decay simultaneously. So it will mostly, for example, superoxide anion is unstable and decays spontaneously to oxygen and hydrogen peroxide in the presence of water. And in addition, we have antioxidants which will either block free radical formation or inactivate free radicals. That is the function of antioxidants. So examples are lipid soluble or fat soluble vitamin E and A, as well as ascorbic acid and glutathione. So iron and copper can catalyze the formation of reactive oxygen species. So usually the reactivity of the iron and copper is usually reduced by binding to storage and transport proteins. So these iron and copper reactivity can be reduced when these metal ions are binded to the specific transport proteins such as transferrin, ferritin, lactoferrin and ceruloplasmin. So these will prevent the metals from participating in reactions that generate ROS. Next, several enzymes act as free radical scavenging systems and break down hydrogen peroxide and superoxide anions. So catalase, as we know that it is present in peroxisomes, so it decomposes hydrogen peroxide. That is the function of catalase. Superoxide dismutase. Superoxide dismutase will convert superoxide anion to hydrogen peroxide in presence of water molecule. So uses both manganese SOD, which is localized in mitochondria, and copper zinc SOD superoxide dismutase, which is found in cytoplasm. So it uses both manganese superoxide dismutase and copper zinc superoxide dismutase. Next we'll see about glutathione peroxidase. Glutathione peroxidase will protect against injury by catalyzing free radical breakdown. That is, this hydrogen peroxide reacts with glutathione homodimer and along with water or 2-hydroxyl free radical. So the intercellular ratio of oxidized glutathione to reduced glutathione reflects the oxidative state of the cell and is an important indicator of cell's ability to detoxify ROS. Is very important. So the ratio of oxidized glutathione to reduced glutathione is important indicator of cell's ability to detoxify reactive oxygen species. Next, let's see about pathological effect of free radicals. First, lipid peroxidation in membrane. See, in presence of oxygen, free radicals will cause peroxidation of lipids within plasma and organelle membranes. This damage is initiated when the double bonds in unsaturated fatty acids of membrane lipids are attacked by oxygen-derived free radicals. So, the double bonds in unsaturated fatty acids of membrane lipids are attacked by oxygen-derived free radicals, particularly by OH radical. So the lipid-free radical interactions will result in formation of peroxides that are unstable and reactive, and an autocatalytic chain reaction ensues called propagation that can result in extensive membrane damage. Next, oxidative modification of protein. So free radicals will help in oxidation of amino acid side chains, formation of covalent protein-protein cross-links like disulfide bonds. Disulfide bond we have seen already in insulin, so which is a peptide hormone. So oxidation of protein backbone. All these will result in damaging the active site of enzymes, disrupt the conformation of structural proteins, and all these will leads to proteasomal degradation of unfolded or misfolded proteins and cause havoc throughout the cell. Next, relations in DNA. So free radicals are capable of causing single stranded and double stranded DNA breaks in DNA. Cross-link, it will what it do is it will cause single and double strand DNA. So it will digest the DNA and it will cross-link the DNA strands. And hence oxidative DNA damage has been implicated in cell aging and also in malignant transformation of cells. Next, let us see about disturbance in calcium homeostasis. See, calcium ions are second messengers in signaling pathways. We know that there is a secondary messengers in signaling pathways, but excess amount of calcium in cytoplasm is an important source of cell injury. Let us see how. So cytosolic free calcium is normally maintained at a very low concentration compared with extracellular levels, and most intracellular calcium is found in mitochondria and endoplasmic reticulum. Histamine and certain toxins will cause excessive increase in cytosolic calcium level because of release from intracellular stores from mitochondria and endoplasmic reticulum, and later due to influx across the plasma membrane. The plasma membrane becomes more permeable to calcium ions. So accumulation of calcium in mitochondria will result in opening of mitochondrial permeability transition pore we have seen earlier, and hence in failure or leads to failure of ATP generation. Increased cytosolic calcium will activate enzymes such as phospholipases which cause membrane damage, proteases which break down both membrane and cytoskeletal proteins, and endonuclease responsible for DNA and chromatin damage, and ATPases that leads to ATP depletion. Next, let's see about endoplasmic reticulum stress. So the accumulation of misfolded proteins in endoplasmic reticulum can stress adaptive mechanism and also trigger apoptosis, which is very fatal to the cells. So the capirones in endoplasmic reticulum will control the proper folding of newly synthesized protein. That is where maybe asked in MCQs, capirones. And then, however, if any unfolded or misfolded proteins accumulate in the endoplasmic reticulum, they trigger a number of alterations, which are called as unfolded protein response. This unfolded protein response will activate signaling pathways that will increase the production of chaperones. Okay, so chaperones in endoplasmic reticulum helps in controlling the proper folding of newly synthesized proteins, and misfolded proteins are sent to the cytoplasm, sent out of the endoplasmic reticulum. But due to this presence of unfolded proteins in mitochondria, they will increase the production of chaperones, and it will enhance the proteasomal degradation of abnormal proteins and slow protein translation. So it thus reduces the load of misfolded proteins in the cell. That is the function of chaperones. But however, in the if the cytoprotective response is However, if this response is unable to mingle with the accumulation of misfolded proteins, the cell will activate caspases and it will induce apoptosis. There is no other way for the cell to escape. This process is called endoplasmic reticulum stress. Okay, uh, so that is what you have seen earlier. Then um, in aging, the increased misfolding may be due to mutations, decreased capacity to correct misfolded proteins by chaperones, which usually occurs in aging. Protein misfolding may also be increased in viral infections. So in viral infections, protein misfolding is increased, and when proteins encoded by viral genome are synthesized in large quantities, that they overwhelm, that is that they cannot overcome the quality control system, and they will ensure that normally usually quality control system ensures normal protein folding. So insulin. So next we'll see about insulin resistant states. See, increased demand for secreted proteins. So insulin resistant states and changes in intracellular pH and redox state or other stresses that result in misfolded protein. That's very important. So insulin resistant states, changes in intracellular pH and redox state or other stresses that result in misfolded protein accumulation. Okay. Okay. So foldases, what they will do is protein misfolding. Before that, protein misfolding is a key indicator for many neurodegenerative diseases to Alzheimer's disease. So foldases usually require ATP to function. So this is not needed, foldases. So that is what we have seen in mechanism of cell injury. This is the summary. You can go through it in Robbins. So thank you. So we will be seeing about clinical pathologic correlations in the next lecture. Thank you.